Hey everyone, and welcome back to The Explainer. So, if you've ever found yourself wondering how you're actually able to make sense of this noisy, chaotic world we live in, you are definitely in the right place. Have you ever thought about how your brain manages to untangle the absolute invisible web of sound crashing into your ears every single second? Well, today we are focusing exclusively on section 5.1 of our source material, which is all about the importance of frequency selectivity. We're going to take a really deep dive into the biological marvel that is human hearing, and we'll discover how our auditory system pulls off what is honestly one of the most incredible sensory magic tricks in all of nature. Let's get into it.
Okay, let's kick things off with a quick thought experiment. Picture this. You're sitting in a grand concert hall, right? The orchestra is playing this massive, beautiful, sweeping symphony. And out of nowhere, you can effortlessly hear the high, piercing trill of a single piccolo, cutting right through the deep, heavy, rumbling groan of a double bass. Now, how is that actually possible? Because in the air between that stage and your ear, those two sounds don't exist separately at all. They have perfectly mixed together into one single, incredibly complicated sound wave. And yet, your brain just instantly unpacks them. To understand exactly how we solve this crazy acoustic puzzle, we actually need to look at a totally different sense first.
Section 1, place-to-place mapping.
Let's establish a bit of a baseline here by looking at how we intuitively understand the world through our visual system. Because our eyes are our dominant sense, we kind of just assume all our senses operate the same way, right? But think about the core mechanism of sight. When you look around the room you're in right now, your visual system is focusing the light coming from each specific point in space onto a very specific corresponding place on your retina. So the physical world is mapped directly onto a spatial arrangement of photoreceptors. It's a very geometric, one-to-one translation of space.
Section 2. Frequency to place mapping.
So if our vision is all about mapping physical space, what on earth is our auditory system doing? Because sound waves definitely do not carry neat little spatial blueprints of a room. Well, while the visual system performs a place-to-place mapping of the world, the auditory system does something entirely different. It performs a frequency-to-place mapping of the acoustic world. Your ear isn't really trying to build a spatial map first and foremost. Instead, it is obsessively focused on pulling apart the sound wave into its individual frequency components. And honestly, the way it pulls this off is just a total masterpiece of biology. Deep, deep inside your inner ear is this structure called the basilar membrane. The basilar membrane essentially performs a partial spectral analysis. Scientists call this a Fourier analysis of the incoming sound. Think of it like a mechanical prism. Just like a glass prism splits white light into a rainbow of individual, distinct colors, the basilar membrane physically splits a complex sound wave into its individual frequencies. Each specific place along that membrane is most sensitive to a totally different frequency component. That, right there, is frequency-to-place mapping in action.
Section 3. 3 cones versus hundreds.
We usually think of our vision as this ultra-high-definition, incredibly detailed sense. But when we actually look under the hood at the biological hardware, a completely different story emerges. Despite our vivid, rich perception of a fully colored world, the visual system effectively only has 3 different patterns of sensitivity to spectral information. Just 3. These correspond to the 3 types of cone cells in your retina. They essentially act like broad bandpass filters for light, which actually gives us a surprisingly limited, compressed picture of true light wavelengths. Now compare that to the auditory system. The human ear has arguably several hundred different spectral sensitivities. That massive contrast is just staggering. While your eye is getting by with just 3 broad filters for color, the basilar membrane in your ear is continuously extracting incredibly precise, highly detailed information about the spectral composition of sounds using hundreds of distinct sensitivities. Your ear isn't just a microphone, it is a high-resolution frequency analyzer.
Section 4. Shapes versus changing spectra.
So you might be wondering, why did evolution design our senses with such a huge discrepancy in spectral resolution? Why does the ear need hundreds of filters when the eye only needs 3? Well, there's actually a perfect functional reason for it. Visual objects are characterized mainly by their static shapes. Spatial information is what tells us where the edge of a cliff is, or the shape of a predator, right? But auditory objects, like a human voice or a musical instrument, are characterized mainly by their spectra, and specifically by the way those spectra shift and change over time.
Let's look at how this applies to everyday human speech. How do you actually tell the difference between the vowel sound E and the vowel sound ah? You do it because your brain is subconsciously tracking formants, which are basically the positions of spectral peaks. Different vowel sounds in speech are identified entirely by where these formants are positioned. And it goes even further than that. The specific way the frequencies of these formants shift and slide over literal milliseconds actually helps your brain identify the consonants that came right before them. It's wildly fast processing.
And by the way, it's the exact same deal with music. Different musical instruments are identified by the specific spectral distribution of their harmonics. This sound quality is what we call timbre. So the fundamental pitch of a note might be the exact same, but the timbre, which is that unique fingerprint of changing spectra, is what lets you instantly know you're hearing a piano instead of a guitar. Pretty much all the meaning we extract from the sounds around us is largely dependent on this incredibly precise spectral information.
Section 5. Separating simultaneous sounds. Now, identifying a single isolated sound is one thing, but the real world is messy, and it is loud. This brings us to the true superpower that this high-resolution frequency selectivity grants us. Let's go all the way back to that thought experiment from the very beginning, the double bass and the piccolo. Most of the acoustic energy of the double bass is heavily concentrated in the low-frequency regions, right? Whereas the energy of the piccolo is all concentrated up in the high-frequency regions.
Because when those two instruments are playing simultaneously on stage, physics dictates that their sound waves literally mix together in the air. They hit your eardrum as a single, combined, totally chaotic sound wave. But here is the magical unmixing process. Because the instruments cover completely different frequency ranges, the basilar membrane steps in as that biological prism we talked about. It physically vibrates at different places for the high piccolo frequencies versus the low double bass frequencies. It just neatly unpacks that combined wave right back into its original separate components.
And get this, our auditory system is so wildly advanced that we can even separate two complex tones that have overlapping harmonics distributed over the exact same frequency region, just as long as their fundamental frequencies are different. Without this acute, hyper-detailed frequency selectivity, every simultaneous sound in the world would just blur together into an incomprehensible wall of noise. And actually, this loss of separation is exactly why listeners with hearing impairment often find it incredibly frustrating to follow a conversation in a crowded, noisy room. The frequency selectivity is what starts to break down.
Section 6. Fundamental to sound perception.
Ultimately, everything we've covered today highlights a profound truth about how we experience the physical world. I want to leave you with this direct quote from the source material which really nails it. Frequency selectivity can be considered just as important to hearing as spatial sensitivity is to vision. It is quite literally the absolute bedrock of how we perceive sounds. So the next time you're sitting in a busy cafe, just effortlessly tuning out the background chatter to focus on the voice of the person across from you, take a second to marvel at the hundreds of spectral filters working continuously in your inner ear. It really makes you wonder, what other hidden biological superpowers are you using right now without even realizing it? Keep learning, stay curious, and we'll see you in the next explainer.